strategies to replace a defective gene in the nervous system. In the second talk, we heard about strategies to neutralize the effect of a toxic gene product in the nervous system. And now for this last talk on genetic approaches to neurological disease, we'll take neither approach, but instead we'll use gene therapy as a tool to deliver a potentially therapeutic protein in a site-specific manner in the nervous system. So I'll be talking to you about gene therapy in neurodegenerative disorders, focusing on Alzheimer's disease, and I'll take you through the beginnings of these clinical programs. And focusing on Alzheimer's disease, I'd like to point out at the outset that what I'll be talking about is an alternative to approaches for Alzheimer's disease that target either amyloid or tau. And that, since it's an additional target for therapy in Alzheimer's disease, it offers the potential down the road of a combination therapy with other therapies should they be shown to be effective in the future. And I'd like to acknowledge that many people have contributed to this program over a number of years. This is basically summarizing 20 years of work and bringing us up now to the present day when we're about to file an IND for a new clinical program. And in terms of targeting neurodegenerative disorders with growth factors, this has been through 3 clinical programs. The first is nerve growth factor gene therapy in Alzheimer's disease through phase 1 and 2 clinical trials that I'll describe for you. Then I'll move on in the second half of the talk to the new clinical program that we're advancing to the clinical arena. There have also been trials of another growth factor in Parkinson's disease through phase 1 and 2 clinical trials that I won't discuss. Those had negative outcomes and are currently in a new iteration with improved gene delivery technology. So why are we talking about nervous system growth factors? So these are a family of about 50 proteins that are normally present in the nervous system during neural development. These help to shape the pattern of neural connectivity and to support neuronal survival during neural development. But in the adult, growth factors continue to be produced in the brain, trafficked through the brain, and they have an important role in maintaining neural circuitry. And indeed, in some neurodegenerative disorders like Alzheimer's disease, growth factor levels decline. That loss of growth factors may be a secondary consequence of cellular degeneration. We don't know that the loss of growth factor availability is causative in Alzheimer's disease. And most importantly then, in terms of therapy, there has been a literature for about 20 years now that when growth factors are delivered as therapeutic proteins in the context of neurodegeneration, that they prevent cell death and stimulate cell function. And growth factors are extracellularly available. When they bind to extracellular receptors on the cell, they induce the phosphorylation of intracellular signaling mechanisms that generally activate PI3 kinase and AKT to counter cellular apoptosis. They also activate a second general stream of signaling through MEK, ERK, and MAP kinase that activates CREB and enhances the functional state of the cell. In neurons, this means that they increase synapses and synaptic transmission, and they counter neuronal death, as I'll show you. So this program is based on the hypothesis that growth factors will prevent neuronal degeneration or death and stimulate the function of remaining neurons in neurodegenerative disorders such as Alzheimer's disease. So the work I'll talk to you about today has been previously published, leading up to a clinical trial now. So this is what growth factors do in the brain. This was the first reported effect of growth factors in the rat brain about 30 years ago. And these results were reported by Franz Hefti, Fred Gage, and others. And basically, this shows the adult rat brain. This is the population of cholinergic neurons in the rat brain labeled for for cholinacetyltransferase. And one can induce the death of these cells on the left side of the brain by cutting their projection to the hippocampus. These cells normally project to the hippocampus and provide it with its most abundant source of acetylcholine. And the function of these inputs is to regulate neuronal excitability. So we can model the death of these cells by cutting their projection to the hippocampus. And when we do it, cells on this left side, the lesion side of the brain, degenerate and die. But when nerve growth factor, the first growth factor identified, is delivered into the brains of these animals, you can see that there appears to be a reduction of cell loss, and indeed the magnitude of cell production consistently in various laboratories independently has been about 80%. So instead of about 75% cell loss, there's about 80% cell survival. And at the time that this biology was identified about 30 years ago, it was really revolutionary because it was the first approach that had been identified that could convert inevitable cell death to extensive neuronal survival. And the implications for the treatment of neurodegeneration were evident. And a number of additional studies followed over the next 5 to 10 years in a variety of injury models showing broadly that NGF prevented the death of cholinergic neurons, stimulated their function, and improved cognition. And we took these observations to the non-human primate brain. We did an experiment similar to what I showed you. We cut the projection of these cholinergic neurons in the monkey to the hippocampus. On the lesion side, they degenerate. Here you have a sense that there's neuroprotection in the presence of nerve growth factor. And when we quantify the effect, there's about 80% cell survival. Once again, when otherwise there's about a 75% loss of neurons. So this neuroprotective ability continues into the non-human primate system. And a number of additional studies came up from various labs showing that if we deliver nerve growth factor, for example, in this case to aged monkeys, with normal age in a monkey, there's a reduction of these cholinergic inputs to the cortex. This is staining for the cholinergic axons as they reach their targets in the cortex. And compared to young monkeys, there is an attenuation. But when nerve growth factor is provided to the aged monkeys, you have a sense that there's protection, and indeed there's a complete restoration of these inputs of cholinergic systems to the cortex. And here's what happens in Alzheimer's disease in humans. With the aged human, there's a little bit of reduction compared to young humans, but in Alzheimer's disease, there's a dramatic acceleration of this loss of cholinergic systems. So the rationale of this program clinically would be to try and address this cholinergic component of neuronal loss in Alzheimer's disease. I'll briefly mention that Alzheimer's disease is the most common neurodegenerative disorder. It affects more than 20 million people worldwide. Those numbers are rapidly increasing, and we still lack effective therapies to prevent degeneration in Alzheimer's disease. And actually, so to make this point, of the pathological substrates, there's a formation of amyloid plaques in the brain. There's abnormal processing of the amyloid precursor protein. There's neurofibrillary degeneration within neurons themselves. These collectively contribute to neuronal toxicity. And these lead first to the loss of synapses in the brain, and then later to the death of neurons in the brain. So this is a progressive disorder. The mean survival is 7 years, and it's untreatable. There are drugs that provide very mild symptomatic benefits, but the benefits are so mild that the drugs are barely worthwhile. So because these cholinergic systems are a component of degeneration, and because we have a growth factor that can prevent the degeneration, the rationale of using nerve growth factor to treat neurodegeneration in Alzheimer's disease was to try and slow this cholinergic component of loss. Now, does this make sense when so many neural systems degenerate in Alzheimer's disease and nerve growth factor only affects one of them? There is a rationale. It's listed here. Basically, the amount of cholinergic deficiency, particularly in early Alzheimer's disease, is one of the strongest correlates of cognitive impairment. So if that held up, if we could target the system and see an improvement in cognition or a delay of decline, that could be useful. But we recognize a priori it would not target all of the neuropathology by any means. At best, it could be a symptomatically beneficial approach. But the Achilles heel of trying to test the ability of growth factors to impact progressive neurodegenerative disorders has been how do we get them into the brain? And there are two constraints. First, these are relatively large and polar proteins, so they don't cross the blood-brain barrier, unlike AAV9. Secondly, if we infuse them centrally and they disseminate broadly through the CNS, they cause intolerable off-target effects that arise from other neural systems that are not degenerating, that respond to the growth factor and upregulate their functional state. And those adverse effects include pain, weight loss from stimulation of hypothalamic satiety receptors. And there's also migration of Schwann cells into the CNS in a layer that encases the brainstem, not something you'd probably want to have done for our neurodegeneration, John, right? And so the problem has been they're biologically potent, but how do we get them into the brain, and how do we restrict their delivery to specific neuronal populations that are degenerating? There are a few options for doing that. One would be to pump the protein into the brain directly into the affected regions, and the other could be gene delivery. And I didn't start life as a gene therapist, but I'm ending life as a gene therapist because this appears to be an effective means of addressing that need to get intraparenchymal delivery, but regionally restricted delivery. So when we began our first human trial of gene therapy in the adult brain for any indication, which was this nerve growth factor program for Alzheimer's disease, we began with ex vivo gene therapy because at that time, 17 years ago, we didn't have AAV vectors. We didn't have vectors that would infect adult non-dividing neurons of the nervous system. So we began with vectors that could infect dividing cells, in this case, patient fibroblasts. They secreted NGF, and then we injected those fibroblasts into this cholinergic nucleus in the brain to see if it would impact neurodegeneration. So there were 8 patients in that early trial, begun in 2001. We injected into 5 sites per side of the brain, and we found that it resulted in no adverse effects, none of these off-target effects of broad NGF dissemination in the nervous system. We saw a significant increase in PET glucose metabolism responses in patient brains, and we saw an unequivocal neuronal response to the growth factor. So that's summarized here. These are first patient PET scans for 2-fluorodeoxyglucose that measures metabolic activity in the brain. These are the mean PET scans of the patient cohort before undergoing NGF gene delivery. I'm losing this pointer, that's why the green comes in and out. But these are the pre-treatment scans, and these are the post-treatment scans, an average of 6 to 8 months later. Typically, there's less red over time as metabolic activity declines. And we saw a reversal of this with a significant increase. So there was this PET surrogate marker. In terms of cognitive decline, this was a very small phase 1 trial. There were no controls, so that, the best we could hope to determine from these first data was just that we weren't harming cognition. And it didn't look like we were harming cognition. This was the rate of cognitive decline in the year before undergoing gene delivery, and one can see there was a reduction in rate of decline in the first year, the 6 to 18 months, and 12 to 24 months. Again, nothing statistically meaningful here other than we didn't harm patients, and this was enough of a rationale to move on to a larger clinical trial. But by this time, the world of gene therapy had caught up in terms of technology for gene delivery into the brain, and we could now move to AAV vectors. So instead of the cumbersome procedure of harvesting patient cells, growing them in cell culture, infecting them with retroviruses, then harvesting the cells and planting them in patients, we could simply inject an AAV vector expressing human NGF into the brain directly. So this results in extended expression periods in the brain for up to 9 years, as I'm about to show you in the human brain. It's simpler than ex vivo gene delivery. These AAV vectors are quite safe. They don't integrate into the host genome, or they do so minimally and haven't been associated with insertional mutagenesis, and they don't appear to result in inflammatory responses when injected into the brain. So there were 10 patients in a phase 1 trial. The findings more or less paralleled what I just showed you for the ex vivo trial, so we moved ahead to a phase 2 multi-center trial, enrolling a total of 49 patients at a number of sites that are listed here. And the last patient was treated in 2015, and the analyses have also been completed, and I'll report those in a moment. But let me just show you first about biological engagement of the target. So this is the site of gene delivery in one of the brains that came subsequently to autopsy, and this is nerve growth factor protein expressed in the brain in the intended target region. And these are the cholinergic neurons that are within the same region, and this again is an NGF label indicating that the cholinergic neurons have expressed the NGF of interest. In sites adjacent to nerve growth factor gene delivery, this is what the cholinergic neurons look like. And this is about an average neuron in this region. At more distant sites in the same brain from NGF delivery, this is the average cell size. It appears to be smaller. And this is an Alzheimer's disease control brain. So this raises the question that there's some hypertrophy of these cells, and when quantified, indeed, there was some hypertrophy of these cells. So there's biological engagement of the target. There's in vivo gene expression and biological engagement. And there's persistent gene expression up to 9 years after gene delivery, shown here in a patient that received AAV2. So here's the NGF gene expression at the site of gene delivery. these are the target neurons, and here they are shown at higher magnification. They're accessing NGF, but note here that this injection site is not in the nucleus. It's off target, and the amount of NGF spread from the injection site is very low. So with that in mind, the phase 2 trial was negative. So we had excellent safety. There was persistent gene expression. There were classic trophic responses predicted by the animal models, but the phase 2 trial was entirely negative. No, whoops, my goodness, that is, okay, here we are. So this is the outcome on this cognitive measure, the mini mental status examination. Higher is better on this scale. These are months after gene delivery. The NGF patients are shown in gray, and the sham surgery control patients are in red, and you can see there's no detectable benefit. There's not a significant worsening either. It's just that the NGF patients started slightly lower than the control group. So there was no benefit. So this was a failed phase 2 trial, and 95% of clinical trials fail. You know, Brian Kaspar has just given us one of the rare exceptions, or what looks to be a rare exception to that rule. And why do so many trials fail? Well, they may fail because the approach is simply biologically ineffective, that there was some problem with the preclinical program leading up to it. But trials can also fail because one fails to engage the target. In this case, there could be inaccurate targeting or spread of the intended viral vector. Clinical trials can fail because the approach is unsafe or because the clinical instruments are insensitive to detect a benefit. Trials can be underpowered, certainly as this was, but we see no hint of an effect. And then there might be insufficient potency of a monotherapy, and one might see a benefit with a combination of therapies. Then finally, there's much more noise in the clinical arena in human trials than there are in animal studies that lead to translation. So these all collectively contribute to this very high failure rate. But in this particular case, let me focus on this problem. We've now received the brains of about 10 patients in the trial, and very few of the injection sites were accurately targeted. Almost all of them were misses. In addition, the vector spread much less than we predicted they would have, based on preclinical data in rodents and primates. So with that collectively, we've learned from failure. One of our greatest lessons, teachers in life, is failure, right? And so we learned the lessons of that and have moved to a new program taking advantage of those lessons. So we've moved on to a new growth factor targeting a different set of neural circuits in the brain. So this was preclinical work advancing while the NGF trial was underway. But this is a more compelling target, and this is what I'll spend my last few minutes on here. And again, a number of people have contributed to this work, particularly those highlighted in green. I'd particularly like to highlight Chris Bankevich at UCSF, who has developed new targeting methods that we'll use in this next clinical program. So in the first program, we were delivering NGF to this basal forebrain cholinergic neuron to affect cholinergic projections throughout the cortex. And there's a good rationale for doing that. In the second program, we're going to use a different growth factor, brain-derived neurotrophic factor, or BDNF. BDNF is the growth factor for cortical and hippocampal neurons. So NGF has no effect on the survival of cortical or hippocampal neurons, but BDNF does. And in preclinical studies, it prevents the death of these neurons and it stimulates their functional state. So we're directly targeting the cortical systems that are dying in Alzheimer's disease, potentially by delivering BDNF. And we did a number of studies that supported the potency of BDNF on these neural circuits. One of the classic models of Alzheimer's disease are transgenic mice that overexpress known human mutations that lead to Alzheimer's disease. These mice have deficits in learning and memory on the Morris water maze. So these are APP transgenic mice. They don't learn as well as wild-type mice. And if we give them a viral vector expressing BDNF, then you can see they have a significant improvement, although they start equally impaired to the APP transgenics. By the end of the testing period, they have learned better. These mice also show an improvement on a second hippocampal-dependent cognitive function, which is hippocampal-dependent fear conditioning. They show improved learning compared to the wild type, or rather compared to the APP transgenics that don't receive BDNF. And BDNF delivery to these APP transgenic mice increases synapses in the entorhinal cortex, the site of gene delivery, and in the hippocampus, a site from which BDNF is transported into the hippocampus. So BDNF delivery to this set of neural architecture significantly restores synaptic density to wild-type mouse levels in the entorhinal cortex and in various regions of the hippocampus, the key neural circuitry that's affected in early stages of Alzheimer's disease. And if we look at the transcriptome of APP transgenic mice compared to wild-type mice, they have a number of downregulated gene sets and some upregulated gene sets, often related to neurodegeneration and neural inflammation. And BDNF gene delivery reverses many of these transcriptional perturbations that occur as a function of the APP mutation. About 2/3 of the aberrant genes are corrected by BDNF gene delivery. But BDNF gene delivery to the entorhinal cortex in these models has no effect on amyloid plaque formation in these mice, so this is apparently occurring independent of effects on amyloid per se, yet influences synapses directly. And if we give this at an early point in life of these APP transgenic mice, there is also a partial neuroprotective effect. This is like a neon show here where the light keeps going out, but there's about a 50% protection from neuronal death in this transgenic mouse model. So we advanced this to additional models, to rat lesion models, aging rat models, and then monkey models. And I'll just, in the interest of time, skip ahead to the non-human primate model. So we can model the degeneration and death of neurons of the entorhinal cortex by cutting their projection into the hippocampus. When we do that, compared to intact monkeys, you see this degeneration of neurons, and there's about a 50% neuron loss in this model, and BDNF protects about 75% of the cells that would otherwise die. And BDNF is injected so a viral vector expressing BDNF is injected into the entorhinal cortex. This is the injection site, and this is BDNF protein being expressed in this target region. And the BDNF is trafficked from that site of injection here into the hippocampus. This is a monkey that doesn't have viral delivery of BDNF into the entorhinal cortex, and this is one that does. You can see there are increases in BDNF protein levels throughout the hippocampus as a function of BDNF transport through the circuitry. In aged monkeys, BDNF induces hypertrophy of these cells. And on a test of learning and memory that's shown here, aged monkeys that receive BDNF show about a 50% improvement in performance. So based on this, we are moving forward to a clinical program, but now trying to use a better method for targeting and confirming gene delivery to this affected brain region. So we target the entorhinal cortex, a small deep structure located here in the brain. And we had targeting problems in our last clinical program that targeted about this region of the brain. So how might we do better? Here's our target seen on the ventral surface of the human brain. So to do this, we're utilizing technology developed by Chris Bankevich at UCSF, and that is now in 3 human clinical trials of gene therapy. And that is to use real-time MR imaging to visualize the needle in the intended target. And we co-infuse the vector with a substance called gadateridol that co-diffuses with the vector. So we can confirm in real time that we have engaged the target and that there's spread of gene delivery to the volume of intended target region. And this shows the visualization of the injection needle in the non-human primate brain. Here's the target down here. And these are serial MRI scans showing that we have effectively engaged the target and that the vector has spread within the target. And this is a movie that shows how this looks in accelerated time. We can infuse both sides of the brain simultaneously. This is the entorhinal cortex, and you can see that this is filled with the gadateridol, which is carrying the AAV2 BDNF vector. And with this improved targeting, we have accurate coherence of the MRI signal with subsequent transduction with BDNF in the non-human primate brain several months later. And one can show this reproducibly. So this is a new targeting approach and represents the next generation, as we see it, of attempting to treat Alzheimer's disease with a candidate therapeutic protein that's biologically powerful, growth factor. So this is, the IND is going to be submitted in this quarter, and then we aim to begin this clinical trial in the first quarter of 2019. So this is an example of a gene therapy program, unlike Brian's example, that hasn't gone well from the start. We've had failures, and we've been doing this clinically for about 17 years now. This is the usual course for the clinical development of new therapies. Monoclonal antibodies had about a 20-year run-up period before they showed their first success. Gene therapy has now shown its first successes in cancer, in retinal degeneration, and very soon, most likely, in spinal muscular atrophy. So we've had this learning curve. And we've had to learn from these experiences that we've had in the human arena too, and recognize that we didn't see a problem with the lack of potency of BDNF at the cellular level in humans. The gene is expressed and the cells are responding, but we haven't targeted. And we hope that now with new methods we can target correctly and finally address the hypothesis that trophic factors will prove useful in preventing cell death and stimulating cell function and ongoing neurodegeneration. And with that, I'll close, and thank you for your attention. Well, we have 1 minute left for questions. Yes?
So, speaking of cell and bias, you know that there are other people who are using stem cells to deliver BDNF. Have you really weighed the two and decided that gene therapy is a better idea, or are you considering both?
Well, you know, stem cell technology is fairly nascent in the clinical arena. It will likely have a run-up time, although I don't know that.
I was thinking microglia, like the people at Irvine.
Yeah, Gene, you know, it's a very interesting question, one that could be explored preclinically, and people have tried that, so it's a worthwhile thought, you bet. Yes?
I was hoping you could discuss your choice of AAV2 as opposed to Dr. Casper's AAV9.
Well, so we need to restrict expression of the growth factor to the region of neurodegeneration. If we have broad expression of BDNF in the nervous system, we'll likely have adverse effects from engaging neurons that aren't degenerating. That's been the experience to date in both preclinical and clinical studies. So we don't want to administer this systemically, and we don't want this to broadly transfect the CNS. So there are a number of vector choices for focal intraparenchymal delivery. 2 is one of many possible vectors. And basically we stuck with 2 because it predominantly infects neurons. It doesn't affect non-neuronal cell types efficiently at all. And it's been safe, and there's an extensive human experience now speaking to the safety of it. So those are the main issues. Okay, thank you again.